Classical sculpture relies on a simple hierarchy, an active artist imposing their will onto inert matter. Stone, bronze, and wood act as silent partners, waiting for human hands to define their form. This model collapses when the material possesses its own pulse. When the medium is biological, the sculptor can no longer treat the material as a passive recipient of shape. Physical sculpting fundamentally changes when the system is alive, actively processing data, and continuously organizing its own structure. This aligns with the theory of vibrant matter, which argues that biological and inorganic materials possess their own inherent agency and capacity for action. Accepting matter as a computational agent forces us to dismantle the historical understanding of sculpture as a finished, static object. Jaime Lobato explored this shift through his 2017 work, Pulcher, Aureus, Filum, a centerpiece of the Naturoficial retrospective. Inside the Centro de Cultura Digital, the work exists in a generative space of darkened galleries and illuminated display cases. A screen on a white pedestal displays shifting red organic patterns that pulse in rhythmic intervals. These patterns are not an animation or a pre-recorded video. They are the functional output of a biological computer powered by genetically modified bacteria. The work redefines the sculptural object. The physical form is the spatial programming language that dictates how the living matter behaves. Lobato's biological medium is E. coli, a rod-shaped bacterium capable of complex collective behavior. By inserting a synthetic genetic circuit into the colony, researchers turned these bacteria into a synchronized population of biological oscillators. The system operates through a mechanism called quorum sensing. As the bacteria grow, they emit signaling molecules that diffuse through their environment. Once the population density reaches a critical threshold, the concentration of these molecules triggers a collective genetic response across the entire colony. This trigger results in synchronized flashes of bioluminescence, making the invisible chemical communication visible to the human eye. The bacterial colony functions as a decentralized network, executing a biological algorithm through its own material presence. The true sculptural container is this microfluidic chip, which houses the active bacteria. The green chambers and the corridors connecting them create a specific geometric layout. This physical architecture restricts the diffusion of chemical signals, acting as the hardware that constrains the system's logic. Modifying the geometry of these chambers changes the travel time for the signaling molecules, which directly alters the temporal rhythm of the bioluminescent flashes. Unlike a traditional sculpture where form is static, here the physical form dictates how the system moves through time. Lobato uses geometry as a spatial programming language, sculpting the behavior of the colony rather than its mass. The artwork encompasses the entire laboratory apparatus, including the instruments required to sequence and maintain the biological samples. Optical sensors first capture the light pulses emitted by the bacteria under the microscope. Computer vision algorithms then translate these optical flashes into digital data. This data is then mapped into the generative audio and visuals that fill the gallery space. This follows Jack Burnham's concept of systems aesthetics, where the focus of art shifts from the object to the relational network of the whole system. The aesthetic experience is found in this continuous chain of translations, where the boundary between scientific tool and art is removed. The installation also tracked a macroscopic experiment, a human choir attempting vocal synchronization. Participants engaged in vocal meditation, using their voices to seek a collective, synchronized frequency. Using fast Fourier transform algorithms, Lobato analyzed the choir's audio to track their progress toward mathematical alignment. The two systems operate on vastly different scales. The bacteria synchronize over a 230-minute cycle, while the human choir aligns in roughly 1 minute. Despite these differences in scale and biology, the underlying mathematical patterns of their synchronization are identical. This specific parity suggests that collective influence and auto-organization are consistent behaviors that bridge the gap between microscopic cells and human participants. The title, Pulcher, Aureus, Filum, translates to a beautiful golden thread. In cybernetics, theorist N. Catherine Hales critiqued the idea that information is a disembodied, immaterial entity. Lobato's installation manifests the physical nature of data. The golden thread is an unbroken chain of heavy material translations, from genetic circuits and chemical diffusion to silicon and light. Every signal requires a body. Information cannot exist without matter, and every act of communication leaves a structural form in its wake. This mathematical link between bacterial and human systems is the core of the natural fissile exhibition. Recognizing these planetary intelligences dissolves the perceived dichotomy between the natural and the artificial. The sculptor's role shifts from a shaper of static mass to an orchestrator of living, active systems. Sculpture becomes a co-constitutive partnership, where human design and the active forces of the biosphere model the work together.